This is an overview of an article entitled Exome Sequencing in Children with Pulmonary Arterial Hypertension Demonstrates Differences Compared to Adults, published in the April 2018 issue of Circulation, Genomic and Precision Medicine. Pulmonary arterial hypertension, or PAH, is a potentially life-threatening disease with a high risk of mortality. It's characterized by pulmonary arterial remodeling, elevated pulmonary arterial pressures, vascular resistance, and subsequent heart failure. PAH can be inherited within families, so-called familial PAH, or it can arise sporadically, so-called idiopathic PAH. In familial PAH, mutations in the BMPR2 encoded bone morphogenetic protein receptor 2, which encodes a member of the transforming growth factor beta or TGF-beta superfamily of receptors, are found in 70% of adult cases. BMPR2 is not the only gene associated with PAH, and mutations in other TGF-beta family member genes, such as ACVRL1, are rare causes of PAH, and other genes such as TBX4, which encodes the TBOX4 protein, have also been implicated. Not just a disease of adults, PAH with pediatric onset is particularly lethal, with a median survival of 10 months following diagnosis. Due to the small number of children relative to adults who are diagnosed with PAH, the genetic underpinnings and the spectrum and frequency of BMPR2 and other gene mutations remains underexplored. Recent advances in the ability of clinicians and scientists to broadly interrogate the genome using high-throughput sequencing methodologies led the authors of the paper under discussion to seek to describe the spectrum and prevalence of genetic variants in pediatric versus adult-onset PAH using whole exome sequencing, or WES. In the paper under discussion, subjects were enrolled at two institutions, Columbia University and Children's Hospital Colorado. Individuals who met World Health Organization criteria for pulmonary hypertension group 1 were included, and probands with both familial and idiopathic PAH were included. Pediatric onset PAH patients were defined as those who were diagnosed at less than 19 years of age. All familial PAH subjects underwent Sanger sequencing-based BMPR2 and ACVRL1 genotyping. Familial PAH subjects who were mutation negative for those two genes, and all idiopathic PAH subjects were assessed with whole exome sequencing. Identified variants were then designated as highly likely to be pathogenic, medium, or low. Within the high pathogenicity group, mutations highly likely to lead to significant alteration to the protein, such as frameshift mutations, were designated LGD for likely gene disrupting. Missense mutations thought to be pathogenic were designated D-miss for damaging missense mutation. The study included a total of 155 and 257 probands with pediatric and adult-onset PAH, respectively. Among familial PAH subjects, 63 out of 104 were found to be positive for a mutation in BMPR2 that was felt to be pathogenic. In addition, following whole exome sequencing of the genotype-negative familial PAH cases and those with idiopathic PAH, 84 of the 350 remaining individuals were found to be BMPR2 mutation positive. There was a similar frequency of BMPR2 mutations in pediatric patients, with 56% familial and 10% idiopathic being mutation positive, compared to adult-onset PAH with 54% familial and 8% idiopathic being positive. LGD mutation positive patients were older at diagnosis than those with D-miss mutations, with average ages of 31 versus 19 years, respectively. In addition to mutations in BMPR2, mutations in a number of other genes were identified, including TBX4. 13 probands harbored novel, unique variants in TBX4. These variants tended to localize specifically to the T-box domain of the protein, shown here in light blue. Pediatric onset idiopathic PAH cases were significantly enriched for TBX4 mutations compared to adult onset patients, 10 out of 130 or 7.7% versus 0% for the latter. There was a trend for a similar association with familial PAH cases. In addition, TBX4 carriers had a 20-year younger age of onset, 7.9 years, when compared to BMPR2 carriers, 28.2 years. The results are depicted here in both panels of the figure. The authors then utilized this WES-generated genetic data to estimate the burden of mutations localizing to all genes associated with PAH. For this, 1,319 unaffected parents from a congenital heart disease cohort in the Pediatric Cardiac Genetics Consortium were utilized as population controls for allele frequency estimates, and only probands of European descent were used. The authors found 2.35-fold enrichment of de novo D-mis variants in the pediatric onset idiopathic PAH cases, and among those, the very early onset cases, that is, those diagnosed at less than or equal to 5 years of age, were enriched for LGD variants. In these pie charts, the overall mutation frequencies for BMPR2 and TBX4 are demonstrated in dark green and light green, respectively, for pediatric or adult onset PAH in either familial or idiopathic PAH. In addition, based on the WES genetic analysis, mutations in a handful of other genes associated with PAH or PAH associated with hereditary hemorrhagic telangiectasia were identified. These mutations made up a very small proportion of individuals, as can be seen in these pie charts. In summary, the authors observed that variants in known PAH risk genes could be found in 70 to 80% of individuals with familial PAH. In contrast, 21% of pediatric onset idiopathic PAH cases were mutation positive. The most commonly mutated gene in PAH was BMPR2. However, mutations in TBX4 were enriched in pediatric patients. There might be genotype-phenotype associations whereby the type of mutation, that is LGD versus D-mis, or the gene locus involved, that is BMPR2 or TBX4, might be associated with different severity of disease or might manifest at different ages of diagnosis. Finally, de novo mutations might explain a substantial proportion of pediatric onset idiopathic PAH cases. Additional research, utilizing large cohorts of children with PAH, might offer the opportunity to uncover additional genes associated with disease and potential new mechanisms of PAH pathogenesis.